Hello, everybody. And now for our final installment of cell reproduction, we're going to talk about meiosis. So in the last lecture, we talked about mitosis, which is cell division in body cells. Okay, so in meiosis, we're going to shift gears. And now we're going to talk about how sex cells or reproductive cells are made in the body. So you're going to recognize some terms, and those are going to be the phases. So in comparison to mitosis, meiosis is going to have two divisions. So mitosis has one, meiosis has two. So you can see prophase one, metaphase one, anaphase one, telophase one, and then you go into prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis. So mitosis, at the end, we had two cells, and at the end of meiosis, we have four. And at the very end of this lecture, we will compare mitosis versus meiosis. So let's go ahead and go right into the phases. So the first phase that we're going to see in meiosis is prophase 1. Now, again, you're going to recognize a lot of similar activities occurring in meiosis in comparison to mitosis. When there is a significant difference, I will let you know. So we're going to see DNA condense. Again, the nuclear membrane is going to dissolve, and our homologous chromosomes are going to pair up. Now, this is different. Okay, so we have homologous chromosomes that are going to pair up, and it's going to be called a tetrad. Okay, so now in comparison to mitosis, we didn't see homologous chromosomes line up. Okay, but in meiosis, we are. And this is where crossing over occurs. Again, let me go back. This is important. So this is another key difference that's happening in meiosis in comparison to mitosis. So what is crossing over? Now, it's very important, and this is where we get our genetic diversity. So this crossing over is one of the reasons why you don't look exactly like your brother or your sister. It's because of this, and it's genetic exchange. So crossing over is the process when those homologous chromosomes form the tetrad, and when they're in that tetrad, they're actually exchanging information. So they're handing this information off and then taking something back, okay? Now, these, again, they're going to exchange genes. So you can kind of see on here we have an X, we have a Y, and we have a Z. So these letters represent genes. So keep in mind that, again, I know I've used this example before, but they're going to exchange genes for, let's say, eye color. Okay, they're not going to exchange a gene for eye color and get hair color in return. Okay, that's not how it works. And if that does work, that's when we start seeing genetic disorders. So in this case, crossing over, let's say we're going to exchange that eye color for an eye color. Now, this will make more sense when we also start talking about genetics and dominant versus recessive. Okay, so we will touch back on crossing over and meiosis when we go over that. But right now, what you need to know is you need to know that crossing over is the exchange of genetic material, and it's going to create genetic diversity amongst reproductive cells. Metaphase 1. Okay, so again, the chromosomes are going to line up randomly. Again, this is going to be another source of genetic variation. So we're going to see those tetrads line up in the middle. Oops, sorry. Another important thing that cells will check in the past lecture, I talked about that mitotic phase. We want to make sure that our chromosomes are lined up on the middle, and this is going to ensure that this chromosome goes this way and this chromosome goes this way. If they're not lined up in the middle, there is a chance that both of these chromosomes could head over to this side, and that's where we get a trisomy, or 3 chromosomes, and over here we'd get a monosomy, where there would only be 1 chromosome. Then we see anaphase. Your homologous chromosomes are going to move away again. Remember, they're going to be attached to the centriole with the spindle fiber to the centromere. They're going to move away. Again, the sister chromatids are still attached. Now we're ending the end of meiosis one. So telophase and cytokinesis. So that nuclear membrane is going to form. That DNA is going to start to uncondense. And as it heads into cytokinesis, that cell is going to divide. So cytokinesis, remember the splitting of the cytoplasm. Here we kind of represent that cleavage furrow. And we have 2 haploid daughter cells. Now, we are not done. We have to head into meiosis 2. Okay, the first stage of meiosis 2 is prophase. So now we have 2 haploid daughter cells. They are genetically different at this point because crossing over has occurred. They've exchanged that information. Again, we're going to see the nuclear membrane dissolve. Kind of goes fast. So you can kind of see the membranes dissolving. Our centrioles are going to start moving to the end. Spindle fibers are going to form and attach to that centromere eventually. So metaphase 2, we have those sister chromatids are going to line up in the center or the middle of the cell. So remember, metaphase means the middle. In anaphase 2, we're going to pull away or to the opposite ends of the cell. So again, spindle fibers attached to the centromeres, spindle fibers attached to the centrioles on the side. Telophase 2 and cytokinesis 2. So that nuclear membrane is going to form, the cell begins to divide, cytokinesis occurs, and 2 haploid daughter cells are formed. Now let's visit that term haploid again. So remember, haploid means it has half of the information needed for a human, or in this case, it would be 23 chromosomes. Remember, a full set is diploid, and in humans, that would be 46 chromosomes. So from there, we're going to talk about spermatogenesis and oogenesis. So spermatogenesis is going to talk about specifically the formation of sperm in males. So again, you're going to start with that first cell. You're going to go through meiosis 1, where you have 2 haploid daughter cells. Then those 2 cells are going to divide again into 4 haploid daughter cells, and then they go through a differentiation process. Okay, so when they differentiate, then they're going to turn into sperm. So every cell in your body goes through a differentiation process. And in this case, to form sperm, it's going to be called spermatogenesis. To form an egg cell, it's going to go through oogenesis. So you have your primary cell. It's then going to divide, okay, into a secondary oocyte and a polar body. Now, what you'll notice about the polar bodies is they're smaller. And what happens is these are kind of cells that are useless. They're not going to turn into an egg. And so what happens is they're actually just broken down and the materials are recycled back into the body. But they're not going to differentiate into an egg cell. Okay, so we're going to see that secondary oocyte also going to separate. They have the polar body, again, useless, going to be recycled. And then we're going to go through a differentiation process and we're going to form an ovum, okay, which will become an egg in a female. And during reproduction, we have sperm and egg, which will eventually come together to create a baby. So these are really helpful diagrams too, kind of comparing the processes of mitosis versus meiosis. So we have, at the beginning of mitosis, we have a cell. It's going to go through mitosis, prophase, metaphase, anaphase, telophase. And I remember the stages by saying PMAT. So prophase, P, metaphase, M, A, anaphase, T, telophase, PMAT. And that helps me remember the order, and it might help you too. So again, we start with 46 chromosomes. We're referring to a human in this case. After mitosis, we have 2 daughter cells that have 46 chromosomes in one cell, 46 chromosomes in the second cell. And they're genetically the same, and they're diploid, meaning they have a complete set, and in this case, a complete set is 2 sets of chromosomes. Meiosis, we're going to start with a diploid cell, 46 chromosomes. We go through meiosis 1, meiosis 2, and at the end we have 4 daughter cells that have one set of information or one set of chromosomes, and they're considered haploid. They have half of the information. So that when, in this case, let's just for example say this is sperm, and this is an egg cell. Sperm cell joins with the egg cell, and how many chromosomes do we have? 46, which is the correct number for humans. Then that one cell that's been created from sperm and egg will continue to divide and to differentiate into a human. It's pretty cool. All right, this is also a really good visual. It's going to show you the difference between mitosis versus meiosis. So over here we have mitosis, so prophase, metaphase, anaphase, into telophase, okay? So remember that PMAT, P-M-A-T. And it also is going to tell you what's going on in each phase as well. In meiosis, again, it's going to be meiosis 1 and meiosis 2. Some really important things about meiosis that differ from mitosis is going to be the crossing over of the tetrads, and then also randomly lining up at the plate in metaphase 1, okay? And then again, the end process or the end product is going to be 4 daughter cells that are haploid. They are also genetically different than what we started with at the beginning. And then the last little table I'm going to show you is also really good. It's comparing and contrasting the two. So divisions. In mitosis, we have 1 division, and we have 2 in meiosis. Independent assortment, we'll talk about independent assortment when we talk about genetics, but it does not happen in mitosis. It does happen in meiosis. Synapsis, this means do the tetrads line up? And in mitosis, they do not, and in meiosis, they do. You might see bivalence. That's just another term for a tetrad, those 2 chromosomes attaching to each other. Crossing over, does it occur? No, because we don't have a synapsis. Yes, it does occur. Outcome, we have 2 cells that are diploid. The use is the body cells. In genetics, are they the same or are they different? In this case, they're going to be the same. In meiosis, we have 4 cells that are haploid, and they're sex cells, they're gametes that will eventually differentiate into sperm or egg. And genetically, they are different. They have a variation, and this is it's important because that's where we get our genetic diversity from. That's why you don't look exactly like your brother, or you don't look exactly like your sister. You're a combination of them. All right, and that is the end of meiosis. I hope you guys have a good grasp on the difference between mitosis versus meiosis. And if you have any questions, you can always email or call me. Thanks, guys.